The story of human evolution is one of the longest and most intricate narratives recorded in the natural world. It spans hundreds of millions of years in its deepest roots, and it is written not in language or memory, but in bone, stone, and the molecular architecture of living cells. To understand how human beings came to exist in the form they occupy today, it is necessary to begin far earlier than the appearance of any creature that would be recognizable as human. It is necessary to begin with the fundamental principles that govern all life on this planet and with the geological and biological conditions that set the stage for the emergence of our particular lineage. Life on Earth has existed for approximately 3.8 billion years, perhaps somewhat longer. The earliest organisms were simple, single-celled forms with no nucleus, this known as prokaryotes. For most of life's history on this planet, these microscopic organisms were the dominant form of biological existence. They colonized the oceans, shaped the atmosphere, and drove the chemical cycles that made more complex life possible. The oxygen that fills the atmosphere today is largely a product of ancient photosynthetic bacteria, organisms that lived and died in vast numbers over billions of years, releasing oxygen as a byproduct of their metabolic processes. Approximately 2.1 billion years ago, a new category of cell appeared, the eukaryotic cell. Eukaryotes are cells with a membrane-bound nucleus, and they're the cellular foundation of all animals, plants, fungi, and many other organisms. The origin of the eukaryotic cell is one of the most significant transitions in the history of life. The most widely accepted explanation for this transition, known as the endosymbiotic theory, holds that eukaryotic cells arose when one type of prokaryotic cell engulfed another, and instead of digesting it, formed a stable, mutually beneficial relationship. The engulfed cell eventually became the mitochondria, the organelle responsible for producing most of a cell's energy. This partnership between two formerly independent organisms became the engine of complex life. Multicellular organisms, life forms made of many cells working in coordination, appeared roughly 600 to 800 million years ago, though the exact timing and the earliest definitive examples remain subjects of ongoing scientific investigation. The emergence of multicellularity allowed for an extraordinary diversification of biological forms. When cells begin to specialize and coordinate with one another, the possibilities for structural complexity and functional sophistication expand enormously. Animals, as a broad category of multicellular life, are characterized by their ability to move, their dependence on consuming other organisms for energy, and by certain shared features of cellular organization. The earliest animals whose fossil remains are well studied belong to the Cambrian period, which began approximately 541 million years ago. This period saw remarkable diversification of animal body plans. Among the organisms that appeared during this time were the early chordates, animals possessing at some point in their development a notochord, which is a flexible rod-like structure that provides support, and a nerve cord running along the back. These early chordates were small, soft-bodied filter feeders, living in ancient seas that bore little resemblance to modern oceans in terms of the life they contained. The vertebrates, animals with a backbone, evolved from within the chordate lineage. The earliest vertebrates were jawless fish that appeared roughly 500 million years ago during the Ordovician period. These creatures were small, often armored, and inhabited shallow marine environments. They lacked the biting jaws that characterize most vertebrates alive today, and instead filtered food from the water or consumed organic material from the sediment. Despite their seemingly primitive nature, these early jawless vertebrates were already possessed of a number of structures that would prove important in the long history leading toward mammals and, eventually, the humans, including a skull, sensory organs, and a brain, however small and simple these structures were in comparison to those of later animals. Jawed vertebrates arose approximately 450 million years ago. The evolution of jaws was an event of tremendous ecological significance. Jaws allowed vertebrates to become active predators in a way that jawless forms could not easily manage. Jaws are modified gill arches, structures that originally served a respiratory function in earlier animals, repurposed over evolutionary time into powerful grasping and chewing mechanisms. With jaws came the opportunity to exploit a wider range of food sources, and jawed vertebrates quickly diversified into a great variety of forms. Among these early jawed fish were the ancestors of all modern vertebrates with jaws, which includes every living species of ray-finned fish, sharks, amphibians, reptiles, birds, and mammals. The move from water to land is another foundational transition in the history leading to humans. Approximately 375 million years ago, during the late Devonian period, certain lobe-finned fishes began to develop the capacity to move and breathe in terrestrial environments. Lobe-finned fishes are a group characterized by the presence of fleshy, muscular fins supported by bones arranged in patterns that are homologous to the limb bones of land animals. This is not coincidental. The limbs of all land vertebrates, including the arms and legs of humans, are directly descended from the fin structures of lobe-finned fishes. Fossils such as Tiktaalik rosei, discovered in the Canadian Arctic and described in the early 21st century, provide a detailed view of what these transitional animals looked like. Tiktaalik had a fish-like body, including scales and fins, but also possessed a neck, ribs, and the bony structure within its fins that closely resembles the arrangement of the upper arm, forearm, and wrist in land vertebrates. The transition to land involved profound anatomical and physiological changes. Animals living in water are buoyed by the medium around them and do not need strong limbs to support their weight. On land, gravity places entirely different demands on the body. The skeletal system must support the full weight of the animal. Breathing must occur through lungs rather than gills. The skin must resist desiccation. The reproduction must be managed in a way that does not require an aquatic environment for the earliest stages of life, or the egg must be protected from drying out by a membrane or shell. These challenges were met gradually over millions of years through the accumulation of small anatomical changes that conferred advantages on individuals possessing them. The descendants of these early land vertebrates diversified into the amphibians, and from within the reptile lineage into the amniotes, animals whose eggs contain a specialized membrane called the amnion that protects the embryo and prevents it from drying out. Amniotes include all living reptiles, birds, and mammals. The amniotic egg was a fundamental innovation that freed vertebrates from dependence on aquatic environments for reproduction, allowing them to colonize drier inland environments that had previously been inaccessible to vertebrate life. Among the early amniotes, two major lineages diverged. One lineage led to the reptiles and eventually to the birds. The other lineage, called the synapsids, led to the mammals. The synapsids can be distinguished from other early amniotes by the structure of the skull, specifically the number and arrangement of openings in the temporal region behind the eye socket. Early synapsids, sometimes informally referred to as pelycosaurs, appeared during the Carboniferous and Permian periods more than 250 million years ago. Some of these creatures, such as Dimetrodon, are often depicted in popular reconstructions of prehistoric life, though they are frequently and incorrectly identified as dinosaurs. Dimetrodon was not a dinosaur, nor was it a reptile in the technical sense. It was a synapsid, and it lived during the Permian period, well before the age of the dinosaurs. From within the synapsid lineage arose the therapsids, a more derived group that showed progressive changes toward the mammalian condition. Over tens of millions of years, therapsid lineages showed increasing development of a secondary palate that separated breathing from eating, changes in the posture of the limbs toward a more upright stance, and modifications to the jaw and ear bones. The jaw is one of the most remarkable case studies in evolutionary transformation. In early synapsids and reptiles, the lower jaw is composed of several bones. Over the course of synapsid evolution, most of these bones gradually reduced in size and eventually migrated to become part of the middle ear in mammals, or where they function as the malleus and incus, two of the three tiny bones that transmit sound vibrations. The dentary bone, which was gradually enlarged over the course of this evolution, became the sole bone of the mammalian lower jaw. This transition is exceptionally well documented in the fossil record, and it provides a clear example of how structures can be repurposed over evolutionary time from one function to another entirely different one. True mammals appeared sometime in the late Triassic period, roughly 225 million years ago. These earliest mammals were small. Most were no larger than a mouse or shrew, and they likely lived in ecological niches that were largely dominated by other types of animals, particularly the archosaurs, which included the lineage that would give rise to dinosaurs. Early mammals were probably active primarily at night, a behavior that would have allowed them to avoid the large diurnal predators of their time. This nocturnal lifestyle may have been associated with the evolution of better hearing and enhanced olfactory ability, as well as with changes in the visual system toward greater sensitivity in low light conditions. The Mesozoic era, often called the Age of Dinosaurs, lasted from approximately 252 million years ago to 66 million years ago. During this long interval, mammals remained relatively small and ecologically marginal. The dominant vertebrates on land were the dinosaurs, a diverse and often large group of archosaurs that occupied a wide range of ecological roles, from small insectivores to enormous herbivores and large predators. Mammals during this period were diversifying quietly, evolving increasingly complex teeth adapted to processing a variety of food types, nursing their young on milk, and refining the physiological and anatomical characteristics that define the class today. Approximately 66 million years ago, at the end of the Cretaceous period, a mass extinction event occurred. This extinction event, associated with the impact of a large extraterrestrial body near what is now the Yucatan Peninsula in Mexico, along with extensive volcanic activity in what is now India, eliminated an estimated 75% of all species on Earth. Non-avian dinosaurs disappeared entirely. Many other groups of organisms, certain types of marine reptiles, many species of ammonites, and a wide variety of invertebrate species also went extinct. The mammals, however, largely survived. Though so did the ancestors of modern birds, which represent a surviving lineage of dinosaurs. In the aftermath of this mass extinction, the surviving mammals entered a world fundamentally different from the one their ancestors had occupied. The ecological roles formerly held by large dinosaurs were now vacant. Over the following millions of years, mammals diversified rapidly and dramatically, filling ecological niches across the full range of terrestrial environments. They evolved into carnivores, herbivores, insectivores, omnivores, burrowers, climbers, swimmers, and flyers. They developed a remarkable variety of body forms, reproductive strategies, and behavioral repertoires. This period of diversification, which occurred during the Paleogene period, the first section of what is commonly called the Age of Mammals, established the major groups of mammals that are recognizable today, including the rodents, bats, whales, ungulates, carnivores, and primates. Primates, the order of mammals to which humans belong, are characterized by a set of features that include a tendency toward large eyes, oriented forward on the face to allow for stereoscopic vision, grasping hands with nails rather than claws on most digits, a relatively large brain in proportion to body size, and a tendency toward slow reproduction with a high degree of parental investment in each offspring. The earliest primates, or animals closely related to the primate lineage, appear in the fossil record approximately 55 to 60 million years ago, shortly after the extinction event that ended the Mesozoic era. These animals were small, and they inhabited forested environments. The grasping hands and feet of primates, along with their forward-facing eyes, are generally interpreted as adaptations to an arboreal way of life, life in the trees, where the ability to accurately judge distances and grip branches securely would have been of great advantage. Over the course of the Eocene and Oligocene epochs, spanning roughly from 56 to 23 million years ago, the primate lineage diversified considerably. The primates of this period included early representatives of the major living divisions of the order. One broad division encompasses the strepsirrhines, which include lemurs, lorises, and related animals. The other division encompasses the haplorhines, which include tarsiers, monkeys, and apes. The haplorhine primates are characterized by several anatomical features, including a relatively larger and more complex brain, a different structure of the nose and upper lip, and a more fully enclosed bony eye socket. Within the haplorhines, the catarrhine primates, which include Old World monkeys and apes, represent the lineage that includes humans. Catarrhines are distinguished from their platyrrhine relatives, the New World monkeys, by a number of anatomical features, including the structure of the nose, the number of premolar teeth, and certain features of the ear region. The split between the New World monkeys and the Old World monkeys and apes occurred approximately 40 million years ago, when South America and Africa were separated by a significant oceanic gap. Or how the ancestors of New World monkeys reached South America from Africa remains a question of ongoing interest. One possibility, supported by fossil evidence, is that small primates rafted across the Atlantic Ocean on naturally occurring mats of vegetation, an improbable journey made possible by the currents of that time and the smaller width of the Atlantic compared to its current extent. The apes, a group that includes gibbons, orangutans, gorillas, chimpanzees, bonobos, and humans, diverged from the Old World monkey lineage approximately 25 to 30 million years ago. The earliest apes lived in Africa, which at that time was a continent of lush, widespread tropical forests. These early apes were diverse in size and form. They were distinguished from monkeys by a number of features, including the absence of a tail in most forms, a broader and flatter rib cage, a more mobile shoulder joint, and differences in the structure of the teeth and skull. The greater mobility of the shoulder joint in apes is thought to be related to patterns of arm hanging and climbing that differ from the running along branches posture more typical of most monkeys. During the Miocene epoch, which lasted from approximately 23 million to 5 million years ago, the apes diversified extensively. This period is sometimes called the Golden Age of Apes because the fossil record from Africa, Europe, and Asia shows a much greater diversity of ape species during this interval than exists today. Modern apes are a much reduced remnant of what was once a far more varied and geographically widespread group. Many of the ape genera and species of the Miocene left no living descendants. Others were ancestral to the modern ape lineages, including the lineage that eventually led to human beings. The climate of the Miocene was significant in shaping the evolutionary trajectory of the ape lineage. During the early and middle Miocene, Africa was warmer and wetter than it is today, and tropical and subtropical forests extended across much of the continent. Apes flourished in this environment. Later in the Miocene, the climate began to cool and dry. Forests contracted, and grasslands and open woodlands expanded in their place. This environmental shift had profound consequences for the primates inhabiting Africa, including for the ancestors of the human lineage. Species that could adapt to more open, less forested environments were favored. Species that could not adapt became extinct or retreated to the remaining forest refuges. The split between the human lineage and the lineage leading to chimpanzees and bonobos, the closest living relatives of humans, occurred sometime between approximately 6 and 8 million years ago, based on both molecular clock estimates and the fossil record. This date range is important to emphasize because it reflects the genuine uncertainty that surrounds this question. The molecular clock, a method of estimating divergence, times based on the accumulated differences in DNA sequences between related species, provides one class of estimate. The fossil record provides another. These two lines of evidence do not always agree precisely, and the exact timing of the human-chimpanzee divergence remains a topic of active scientific investigation. It is important to be clear about what the concept of divergence means in this context. When scientists say that the human lineage and the chimpanzee lineage split approximately 6 to 8 million years ago, they do not mean that humans descended from chimpanzees. Modern chimpanzees and modern humans share a common ancestor, a population of apes that lived millions of years ago, and from that ancestral population, two separate lineages evolved independently. One of these lineages gave rise to modern chimpanzees and bonobos. The other gave rise to the various members of the human evolutionary lineage, collectively known as hominins, and ultimately to modern humans. Both lineages have changed over those millions of years, and neither lineage remained static. The earliest hominins, members of the human side of this evolutionary split, are known from fragmentary fossil evidence. Among the earliest candidates for hominin status is Sahelanthropus chadensis, a species known from a skull and some jaw and tooth fragments discovered in Chad in Central Africa, dating to approximately 6 to 7 million years ago. The facial structure of this specimen has features that suggest a relationship to the human lineage rather than to the lineage leading to chimpanzees, and some researchers have interpreted the position of the foramen magnum, the opening at the base of the skull through which the spinal cord passes, as consistent with an upright or partially upright posture. However, the fragmentary nature of the evidence makes definitive conclusions difficult. Another early hominin candidate is Auroran tugenensis, known from fossil remains discovered in Kenya dating to approximately 6 million years ago. These fossils include femur bones, whose internal structure has been interpreted by some researchers as consistent with bipedal walking. Bipedalism, habitual upright walking on two legs, is one of the defining features of the human lineage, and its presence or absence in early hominin specimens is therefore of considerable significance. A somewhat better-known early hominin is Ardipithecus, with fossils from Ethiopia dating to between approximately 4.4 and 5.8 million years ago. The genus Ardipithecus includes two species, Ardipithecus cadaba and Ardipithecus ramidus. The latter species is known from a more complete set of fossil material, including the partial skeleton of a single individual described in the scientific literature in 2009 after years of careful study and preparation of the fragmentary remains. This individual, often referred to by the nickname Ardi, provides a revealing snapshot of what a very early hominin looked like. Ardi was approximately 120 centimeters tall and weighed an estimated 50 kilograms. The skeleton shows evidence that Ardi was capable of walking upright on the ground, but also that she retained significant arboreal capability. Her feet had a grasping big toe that would have been useful for climbing, while her pelvis showed modifications consistent with a degree of bipedal walking. Ardi's brain was small, roughly the size of that of a modern chimpanzee. Her teeth were relatively small and lacked the large canines seen in many other apes, suggesting a diet quite different from that of a chimpanzee. The picture that emerges from Ardi is of an animal that was neither a modern human nor a modern chimpanzee, but something genuinely different from both. An early member of the human lineage, retaining many features associated with arboreal life while already beginning to show the modifications associated with terrestrial bipedalism. The Australopithecines are a group of hominins that followed the earliest members of the human lineage. They lived primarily in Africa between approximately 4 million and 2 million years ago, though some lineages persisted until as recently as perhaps 1 million years ago. The name Australopithecine refers to their discovery in the southern part of Africa, though they are now known from sites across eastern and central Africa as well. The Australopithecines are characterized by a combination of features that distinguishes them both from the earlier, more ape-like hominins, and from the later members of the human lineage. They walked bipedally on the ground and possessed hominid-like teeth, but they retained relatively small brains, small body sizes, and features of the skeleton reflecting continued capability for arboreal activity. The most famous of all Australopithecines is Australopithecus afarensis, known from numerous fossils from sites in Ethiopia and Tanzania, dating to between approximately 3.7 and 3.0 million years ago. This species is represented by one of the most celebrated specimens in the history of paleoanthropology, the partial skeleton of a female individual discovered in the Afar region of Ethiopia in 1974. Given the scientific designation AL288-1 and known more widely by the name Lucy. Lucy was small, approximately 107 centimeters tall and weighing perhaps 29 kilograms, and her skeleton clearly shows the combination of bipedal and arboreal features that characterizes the Australopithecines. Her lower limbs show clear adaptations to upright walking, with a femur angled in toward the midline in a way that places the foot beneath the center of gravity during walking. Her upper limbs are relatively long, and her shoulder and finger bones retain features suggesting significant capability for climbing. Her brain volume, estimated from the size of the skull, was approximately 400 to 500 cubic centimeters, similar to that of a modern chimpanzee and roughly one-third the volume of a modern human brain. The Australopithecus afarensis is also represented by a remarkable set of fossilized footprints discovered at Laetoli in Tanzania in 1978. These footprints, preserved in volcanic ash that fell and then hardened some 3.6 million years ago, show the tracks of at least 3 individuals walking bipedally. The footprints are fully modern in their form. They show an arched foot with a non-divergent big toe, heel strike, and the gait pattern of an upright walking animal. They leave no doubt that by 3.6 million years ago, hominins were walking in a fully or nearly fully modern fashion across the landscape of eastern Africa. Several other species of Australopithecines are known from the fossil record, showing a variety of body sizes, facial structures, and dietary adaptations. Australopithecus africanus, discovered in South Africa and described by Raymond Dart in 1925, is the species for which the term Australopithecine was coined. Australopithecus sediba, discovered at Malapa in South Africa and described in 2010, is a later Australopithecine with some features that have been interpreted as placing it closer to the genus Homo than other known Australopithecines, though its exact place in the human family tree remains debated. Australopithecus garhi, known from Ethiopia and dating to approximately 2.5 million years ago, was found near animal bones that appear to have been processed for their meat and marrow, providing some of the earliest possible evidence for hominin meat consumption and tool use. And alongside the more gracile Australopithecines, those of more slender build, there also evolved a group of hominins known as the robust Australopithecines, or Paranthropines, now commonly placed in the genus Paranthropus. The robust Australopithecines had very large cheekbones, enormous chewing muscles evidenced by a pronounced bony crest running along the top of the skull, and very large back teeth. These features are generally interpreted as adaptations to a diet consisting of hard or tough plant material, seeds, roots, tubers, and similar foods. 3 species of robust Australopithecines are recognized: Paranthropus aethiopicus, Paranthropus boisei, and Paranthropus robustus. The robust Australopithecines represent a side branch of the human family tree. They are hominins, and they shared Africa with the ancestors of modern humans for a considerable period, but they left no living descendants and eventually went extinct. The Australopithecine grade represents a crucial phase in human evolutionary history. During this period, the essential shift to habitual bipedalism was consolidated, and the foundations were laid for the subsequent evolution of a larger brain and more complex behavior. But the Australopithecines themselves, despite their importance in the evolutionary story, were not humans in any meaningful sense. Their brains were small, their stone tool use, if present at all, was minimal and simple, and their way of life was in many ways more similar to that of other large African primates than to anything recognizably human. The emergence of the genus Homo, the genus to which modern humans belong, marks a significant transition in the hominin lineage. The earliest members of genus Homo appear in the fossil record approximately 2.5 to 2.8 million years ago, at a time when the African climate was experiencing increasing aridity and the expansion of open grassland environments at the expense of woodland. This environmental context is thought to have been a significant driver of evolutionary change in the hominin lineage, favoring individuals capable of exploiting a wider range of food sources, including meat from large animals, and of moving efficiently across open terrain. The first stone tools in the archaeological record, belonging to what is called the Oldowan tool industry, appear at roughly the same time as the earliest Homo fossils, approximately 2.6 million years ago. Oldowan tools are relatively simple. They are made by striking one stone against another to remove flakes, producing sharp-edged flakes that could be used for cutting and core tools that may have been used for various purposes. The simple appearance of these tools belies the cognitive and motor skill required to produce them. Experimental studies have shown that making even simple stone tools requires a specific understanding of the properties of stone and considerable practice to execute successfully. The appearance of systematic stone tool production in the archaeological record is therefore generally interpreted as reflecting a significant cognitive development in the hominin lineage. The species most commonly associated with the earliest stages of the genus Homo is Homo habilis, now described from fossils from Olduvai Gorge in Tanzania. Homo habilis, whose name means handyman, had a somewhat larger brain than the Australopithecines, averaging approximately 600 cubic centimeters, and a lighter build of the face and teeth. However, the species also retained many Australopithecine-like features, and its precise relationship to later Homo species, and indeed the question of whether it truly belongs in the genus Homo at all, is a matter of ongoing debate among paleoanthropologists. Another early Homo species, Homo rudolfensis, known from Kenya and dating to approximately 1.9 million years ago, had an even larger brain and flatter face, and the relationship between these two early Homo species, and between either of them and later Homo, remains incompletely resolved. Homo ergaster, known from East Africa and dating to approximately 1.9 to 1.4 million years ago, represents a more significant step in human evolution. This species is closely related to, and by some researchers considered identical to, Homo erectus, the first hominin species to spread out of Africa. Homo ergaster had a substantially larger brain than Homo habilis, averaging approximately 850 cubic centimeters, a taller and more slender body with limb proportions closely resembling those of modern humans, and a reduced size difference between males and females compared to Australopithecines. The best-known specimen of this species is the Turkana boy, a nearly complete skeleton of a young male individual discovered near Lake Turkana in Kenya in 1984. The Turkana boy was approximately 8 to 12 years old at death. The precise estimate depends on assumptions about his developmental rate, and had he reached adulthood, he would have been approximately 185 centimeters tall, a stature within the modern human range. His skeleton is the most complete of any early hominin, and it has provided an extraordinary wealth of information about the anatomy, growth, and possible biology of his species. Homo ergaster was associated with a more sophisticated stone tool technology known as the Acheulean industry. Acheulean tools are characterized by large, symmetrical, teardrop-shaped hand axes that required considerably more skill and forethought to produce than Oldowan tools. The production of an Acheulean hand axe involves removing flakes systematically from both sides of a stone core to produce a tool with a specific, planned shape, a process that involves holding a mental template of the desired end product and executing a sequence of controlled strikes to achieve it. The Acheulean tool industry persisted, with gradual refinements for well over 1,000,000 years, an extraordinary duration that reflects both the effectiveness of this technology and possibly certain constraints on the rate of cultural innovation in early Homo populations. The biology of Homo ergaster also showed changes consistent with an increasingly active, wide-ranging way of life. The long legs and efficient bipedal gait of this species would have allowed sustained walking and perhaps running over long distances, which would have been advantageous both for following migrating animal herds and for ranging across the mosaic of habitats that characterize the African savanna landscape. Evidence from the skeleton suggests that Homo ergaster may have had a more human-like pattern of sweating and thermoregulation than earlier hominins, an adaptation that would have allowed activity during the heat of the day when large predators tend to rest, and that may have been accompanied by a reduction in the dense body hair covering seen in other apes. Homo erectus, closely related to or perhaps synonymous with Homo ergaster, is particularly significant as the first hominin species known to have left Africa and colonized other parts of the world. Fossils of Homo erectus have been found in Java in Indonesia and in China, with the earliest Asian specimens dating to approximately 1.8 to 1.9 million years ago. This means that hominins had reached East Asia within a geologically short time of the appearance of the first large-brained, long-legged Homo species in Africa. The spread of hominins into Asia represents a fundamental expansion of the geographic range of the human lineage, made possible by the anatomical and behavioral capabilities of this species. The environment through which early Homo erectus populations spread was different from today. During the early Pleistocene, the geological epoch beginning approximately 2.6 million years ago, the world was generally cooler than it had been during the Miocene, and the climate was cycling through intervals of glacial advance and retreat. During cold intervals, sea levels dropped as water was locked up in glaciers, exposing land bridges that connected land masses otherwise separated by shallow seas. These land bridges would have facilitated the spread of animals, including hominins, across regions that are now separated by water. The region we now call the Middle East served as a corridor between Africa and Asia, and it was almost certainly through this corridor that early Homo populations first moved from Africa into the rest of the world. The period stretching from the earliest Australopithecines to the colonization of Asia by Homo erectus encompasses roughly 4 million years of hominin evolution. During this time, the lineage underwent fundamental changes in posture, locomotion, body size, brain size, diet, and tool use. These changes did not occur suddenly or in isolation. They represent the cumulative effect of natural selection operating over enormous spans of time on populations of animals living in variable and often challenging environments. Each small anatomical change that conferred a survival or reproductive advantage on an individual was slightly more likely to be passed on to the next generation than changes that did not confer such advantages. Over thousands of generations, these small advantages accumulated into the substantial differences that separate early hominins from their ancestors and from their contemporaries. Natural selection is not the only evolutionary mechanism relevant to understanding human origins. Genetic drift, random changes in the frequency of genetic variants within a population, plays an important role, most particularly in small populations where chance events can have a significant effect on the gene pool. Gene flow, the movement of genes between populations through the migration and interbreeding of individuals, is also important, and it is increasingly clear from ancient DNA studies that the human evolutionary story involves a considerable amount of interbreeding between different hominin populations. Sexual selection, the differential reproduction of individuals based on traits that influence their attractiveness to potential mates, may also have been significant in shaping some features of the hominin body and behavior over time. The environment of African hominins during the period covered in this first part of our exploration was not static. The climate of Africa over the past 5 million years has been highly variable and influenced by global trends toward cooling and increasing aridity, by the long-term cycles of glacial and interglacial periods driven by variations in the Earth's orbit and axial orientation, and by more local factors including the uplift of the East African Rift, which has significantly altered precipitation patterns across the region. The fluctuating nature of the environment would have repeatedly created conditions that favored different combinations of traits in hominin populations, sometimes favoring adaptations to open grasslands, sometimes to closed woodlands, sometimes to the exploitation of lakeshore or riverine resources. This environmental variability is thought by some researchers to have been an important driver of brain expansion and behavioral flexibility in the hominin lineage. Because populations living in variable environments that change unpredictably over time may benefit more from general cognitive flexibility than from highly specialized adaptations to a single type of environment. The African continent of 2 to 3 million years ago was home to a variety of large mammals, including numerous species of antelope, elephant, rhinoceros, hippopotamus, and predators such as saber-toothed cats and giant hyenas. Hominins lived within this ecosystem and were subject to the same ecological constraints as other animals. They needed to find sufficient food, avoid predation, and successfully reproduce. The evidence suggests that early Homo was an opportunistic omnivore, consuming plant foods of various kinds as well as obtaining meat and marrow from animals, either through active hunting or through scavenging from the kills of other predators. The question of the relative importance of hunting versus scavenging in the diet of early Homo has been extensively debated. The current consensus tends to favor a picture in which early Homo was primarily a scavenger of marrow and other secondary carcass products, while later, more cognitively advanced hominins were capable of organized hunting. The social life of early hominins is not directly observable from the fossil record, but certain inferences can be made from comparative biology, from the archaeological record, and from what is known about the behavior of the closest living relatives of humans. The small size difference between males and females in Homo ergaster, compared to the more pronounced size difference in Australopithecines and many other primates, suggests a social organization somewhat different from the harem-based systems seen in some great apes. Among living primates, reduced male-female size difference tends to be associated with more monogamous or pair-bonded social structures, though this relationship is not absolute. Group living would have conferred significant advantages for early hominins, including protection from predators, cooperative foraging, and the sharing of knowledge about resources in the environment. The first part of the human evolutionary story, then, extends from the deepest roots of vertebrate life through the age of the Australopithecines and into the emergence and early spread of the genus Homo. It is a story of gradual but ultimately transformative change, driven by natural selection and constrained by the physical and biological environment, playing out across millions of years on a continent whose landscapes were themselves in constant flux. The foundations laid during this period, upright walking, enlarged brains, tool use, extended ranges, and flexible diets, would support all subsequent developments in the human lineage. The stage is set, and the next chapter of this story involves the deepening of these trends and the increasing complexity of hominin life across multiple continents over more than 1 million years. The second phase of human evolutionary history is, in many ways, the longest and most complex. It encompasses a period of roughly 1.8 million years, stretching from the early dispersal of Homo erectus out of Africa through the emergence of anatomically modern humans. During this interval, multiple hominin lineages existed simultaneously in different parts of the world. The brain underwent its most dramatic expansion in size and complexity. Technology became progressively more sophisticated, and the first indications of symbolic and social behavior began to appear. This phase is characterized not by a single linear progression from less to more advanced, but by a branching, complex pattern of diversification, regional differentiation, extinction, and occasional interbreeding among different hominin populations. Homo erectus, as noted in the preceding section, colonized large portions of Asia by approximately 1.8 million years ago. The Asian populations of this species occupied a wide range of environments, from the tropical forests of Java to the more temperate environments of northern China. The Javanese specimens, known from the sites of Trinil and Sangiran, include some of the most historically important hominin fossils ever discovered, as it was the remains from Java that convinced the scientific community in the late 19th and early 20th centuries that human evolution had indeed taken place, and that the process had occurred across a much longer time span than previously imagined. The Chinese specimens from the cave site of Zhoukoudian near Beijing include evidence not only of the anatomy of these hominins, but also of their behavior, including the use of fire, the consumption of a wide variety of animal species, and the accumulation of remains over tens of thousands of years, suggesting repeated use of the same shelter. The use of fire is one of the most significant behavioral developments in the entire history of the human lineage. Fire provides warmth, protection from predators, and, most importantly from an evolutionary standpoint, the ability to cook food. The cooking of food changes its chemistry in ways that make it considerably easier to digest and that increase the caloric yield that can be extracted from a given quantity of food. Cooking also kills parasites and pathogens that may be present in raw meat. The biologist Richard Wrangham has argued that the adoption of cooking was a critical enabling factor in the evolution of the large human brain, because the energetic demands of a large brain are very high, and cooking made it possible to sustain that energetic burden without a corresponding increase in the size of the digestive system. While the timing of when hominins first habitually used fire is debated, with evidence from some sites suggesting fire use as early as 1 million years ago or more, and more unambiguous evidence for regular hearth use appearing considerably later. The association of fire with Homo erectus at Zhoukoudian, dating to roughly 400,000 to 700,000 years ago, is among the earlier secure evidence for its use. The persistence of Homo erectus in Asia over a remarkable span of time, the latest Javanese specimens may date to as recently as 100,000 to 50,000 years ago, though these late dates remain debated, represents one of the most geographically and temporally extended chapters in hominin history. Over this enormous time span, Asian Homo erectus populations showed both continuity and regional variation. They continued to use stone tools, exploit animal resources, and inhabit a variety of environments, but they did not show the same degree of technological and cognitive change that is documented in the African and European fossil records over the same interval. This contrast between stasis in some regions and change in others is an important feature of hominin evolution during this period. In Africa and Europe, the period between approximately 1.8 million and 300,000 years ago is characterized by a gradual but significant increase in brain size and in the complexity of tool technology. The African and European hominins of this period are often assigned to a broad grouping sometimes called archaic Homo sapiens, or alternatively placed in species such as Homo heidelbergensis, a species named for a jawbone discovered near Heidelberg, Germany in 1907. Homo heidelbergensis is known from fossils in Africa, Europe, and possibly Asia, dating to roughly 700,000 to 200,000 years ago. These hominins had brain sizes ranging from approximately 1,100 to 1,400 cubic centimeters, overlapping with the lower end of the modern human range, and showed progressive refinement of stone tool technology. They were large-bodied, robust individuals, brachiosaurs. And there is evidence from certain sites that they were capable hunters of large game, including horses and rhinoceroses. The fossils from the site of Atapuerca in northern Spain are particularly informative about the biology and behavior of European hominins during this period. The Cima de los Huesos, the pit of bones, at Atapuerca has yielded hundreds of hominin bone fragments belonging to at least 28 individuals, dating to approximately 430,000 years ago. These individuals show a mixture of features, some resembling those of Homo heidelbergensis, others anticipating the anatomy of the Neanderthals who would later inhabit Europe. Remarkably, many of the bones at the Cima de los Huesos appear to have been deliberately deposited there, rather than accumulated by predators or water action, suggesting that the individuals who deposited them had some conception of what to do with the bodies of their dead, a behavior with potential social or symbolic dimensions. Genetic analysis of material from Cima de los Huesos has provided DNA sequences confirming a relationship between these individuals and the Neanderthal lineage. The Neanderthals are among the best known of all extinct hominin species, owing both to the abundance of their fossil remains and to the long history of scientific and public fascination with them. Homo neanderthalensis, the scientific name for the Neanderthals, inhabited Europe and Western Asia from approximately 400,000 to 40,000 years ago. The Neanderthals were stocky, robustly built hominins with large brains, on average slightly larger in volume than those of modern humans, and a distinctive suite of anatomical features, including a large projecting mid-face, heavy brow ridges, a receding chin, large front teeth, and a broad barrel-shaped chest. These features are thought to reflect adaptations to the cold, often glacial environments of Ice Age Europe. The large nose and sinuses of Neanderthals may have helped warm and humidify cold air before it reached the lungs. The broad, muscular body with relatively short limbs would have minimized heat loss compared to a tall, slender body plan. Neanderthals were highly capable hominins with a well-documented archaeological record. They produced a stone tool technology known as the mousterian, named for the site of Le Moustier in France, that involved the careful preparation of stone cores to produce flakes of a specific shape and size. This technique, known as the Levallois method, required planning and foresight, as the shape of the final tool was determined in large part by how the core was prepared before the final flake was struck. Neanderthals used these tools for a variety of purposes, including butchering animals, processing plant material, and working hide. They also used hafted tools in which a stone implement is attached to a wooden handle or shaft, which significantly improves the efficiency with which tools can be used for tasks such as cutting and spearing. Neanderthals were accomplished hunters of large game. The isotopic analysis of Neanderthal bones, which preserves a chemical signature of the foods an individual consumed during their lifetime, consistently shows very high levels of animal protein in their diet, comparable to those of top carnivores such as wolves. Neanderthals hunted mammoths, woolly rhinoceroses, horses, bison, deer, and a variety of other large mammals. They likely used ambush strategies, getting close to large animals before using thrusting spears wielded at close range rather than thrown, to kill them. The robust build of Neanderthal skeletons and the high frequency of healed injuries on their bones are consistent with a lifestyle involving close-range contact with large, dangerous prey animals. Neanderthals buried their dead, at least on some occasions, making them among the very few non-human species in the fossil record for which burial behavior is documented. Sites such as Shanidar Cave in Iraq contain Neanderthal skeletons in postures that suggest deliberate interment. The Shanidar IV skeleton was famously associated with large quantities of pollen in the sediment around the body, initially interpreted as evidence of flower burial. This interpretation has been questioned, and the pollen may have accumulated through other means, but the general evidence for deliberate burial at multiple Neanderthal sites remains. Whether Neanderthal burial behavior reflected symbolic or spiritual conceptions, or was motivated purely by practical or hygienic considerations, cannot be determined from the skeletal evidence alone. There is also evidence that Neanderthals used pigments, made ornaments, and engaged in other behaviors sometimes classified as symbolic or cognitively complex. Ochre, a red iron oxide pigment, has been found at some Neanderthal sites, and there are claims of shell ornaments and feather-decorated objects associated with Neanderthal remains, though the interpretation of these findings as evidence of symbolic behavior is not universally accepted among researchers. Some argue that these behaviors represent independent Neanderthal developments. Others suggest they may have been adopted through contact with modern human populations. The Neanderthals are now known, through ancient DNA analysis, to have interbred with the ancestors of modern humans. When the Neanderthal genome was first sequenced and compared to the genomes of living people in 2010, it became clear that people of non-African ancestry carry a small but detectable percentage of Neanderthal DNA, typically between 1 and 4%. This finding has been replicated and refined by subsequent studies, and it is now well established that the ancestors of modern non-African peoples interbred with Neanderthals on at least one and probably several occasions during the period when modern humans were spreading out of Africa and through the regions where Neanderthals lived. Some of the Neanderthal DNA segments that have been retained in modern human genomes appear to have conferred functional benefits, including some variants involved in immune function, while others may have been slightly deleterious and were gradually removed from the gene pool by natural selection over subsequent generations. In parallel with the evolutionary history of Neanderthals in Europe and Western Asia, a separate archaic hominin lineage inhabited the regions of Central and Eastern Asia that are now China, Mongolia, and Siberia. This lineage is known as the Denisovans, a name derived from Denisova Cave in the Altai Mountains of Siberia, where their remains were first identified through ancient DNA analysis in 2010. The Denisovans are an extraordinary discovery, that they were identified entirely through genetic analysis. The physical remains associated with the first Denisovan DNA consisted of a single finger bone and a tooth. Their anatomical characteristics therefore remained largely unknown until additional fossils were tentatively associated with the Denisovan lineage on the basis of molecular evidence. A jawbone from Siahi in Tibet, found in 1980 but not identified as a Denisovan fossil until protein analysis was conducted in 2019, suggests that Denisovans were robust hominins with large teeth and that they inhabited high-altitude environments as well as lowland regions. The Denisovans interbred with both Neanderthals. The Denisova cave itself contains evidence of individuals who were the offspring of a Neanderthal mother and a Denisovan father, and with the ancestors of modern humans. Modern populations of Melanesians, Aboriginal Australians, and some other peoples of Southeast Asia and the Pacific carry a proportion of Denisovan ancestry, approximately 3 to 6% on average in some populations, reflecting interbreeding between their ancestors and Denisovan populations during the spread of modern humans through Asia. Some Denisovan DNA segments found in the genomes of modern Tibetans appear to have conferred an advantage in high-altitude environments, including variants related to hemoglobin regulation that help people live and work at elevations where oxygen is scarce. The discovery of the Denisovans illustrates the power of ancient DNA analysis as a tool for reconstructing human evolutionary history. Before the genomic revolution, the hominin fossil record was interpreted primarily on the basis of anatomical features and inferences about the relationships between different fossil populations depended on assessments of which skeletal features were shared and which were different. This approach provided valuable information but was limited by the fragmentary nature of many fossil specimens and by the difficulty of determining which anatomical similarities reflected close evolutionary relationship versus convergent adaptation to similar environments. Ancient DNA analysis bypasses these limitations in many cases, providing direct evidence of genetic relationship, and through analysis of differences in the DNA sequence, providing estimates of the timing of population separations and the degree of gene flow between populations. Back in Africa, the lineage leading directly to modern humans was undergoing changes that would ultimately prove decisive for the subsequent history of the species. The hominins of Middle Pleistocene Africa, roughly 800,000 to 300,000 years ago, showed a progressive increase in brain size and an increasing sophistication of technology and behavior. The Middle Stone Age of Africa, which began approximately 300,000 years ago and continued until roughly 30,000 to 40,000 years ago in some regions, is associated with a range of technological and behavioral innovations that represent a significant advance over the earlier Acheulean. Middle Stone Age toolkits included prepared core technology similar to the Levallois method of the Neanderthals, as well as a variety of specialized tools made from carefully selected raw materials, including small, sharp flakes that could be used as the tips of projectile weapons. This last innovation, the use of projectile weapons, is significant because it allows hunters to kill large animals from a safe distance, reducing the risk of injury compared to close-range thrusting weapons. The site of Pinnacle Point in South Africa has provided evidence for complex behaviors in Middle Stone Age populations dating to approximately 165,000 years ago. These behaviors include the use of marine shellfish as food, a resource that requires knowing the tidal cycle and planning visits to the coast at times when shellfish are accessible, the use of ochre pigment, and the production of blade tools through heat treatment of silkrete, a type of stone that becomes easier to work when it has been carefully heated and cooled. The heat treatment of stone for toolmaking requires detailed knowledge of the properties of the material and the ability to plan and execute a multi-step process. It is not a simple or accidental discovery, but a form of applied knowledge that was presumably transmitted between individuals within a social group. Another important African site from this period is Blombos Cave, also in South Africa, which has yielded evidence for a range of behaviors including the production of ochre-based pigment mixtures stored in shells, the engraving of geometric patterns on pieces of ochre, the collection and possibly use of shells as personal ornaments, and the production of finely made bone tools. These objects, dating to between approximately 100,000 and 75,000 years ago, these are among the earliest evidence for what researchers term modern behavior. Behaviors that require symbolic thinking, the use of culturally transmitted information, and the production of objects whose value lies partly in their meaning rather than purely in their physical utility. The emergence of anatomically modern humans, Homo sapiens in the form that would be recognizable as human today, occurred in Africa. The oldest well-attested fossils of Homo sapiens come from the site of Jebel Irhoud in Morocco, described in 2017, and date to approximately 300,000 years ago. These individuals had brains of modern size and a facial structure largely within the modern range, though their brain cases were somewhat more elongated and flatter than those typical of most living people, suggesting that the final evolutionary changes affecting the shape of the brain were still ongoing. Fossils from sites such as Omo Kibish in Ethiopia and Herto, also in Ethiopia, dating to approximately 195,000 and 160,000 years ago respectively, show modern humans with brain cases already closely resembling those of living people. The timing of the origin of modern humans in Africa and the subsequent spread of modern human populations out of Africa and across the globe is a topic that has been extensively studied and debated. The prevailing model, supported by both genetic and fossil evidence, holds that modern humans originated in Africa, that the African population was the source of all living human populations, and that modern humans spread from Africa into other parts of the world beginning roughly 60,000 to 100,000 years ago. or possibly earlier in some cases. This model is sometimes called the recent African origin model or the out of Africa model. An older alternative model, the multi-regional evolution hypothesis, proposed that modern humans arose simultaneously in different parts of the world through the parallel evolution of regional archaic Homo populations connected by ongoing gene flow. The weight of genetic evidence, and particularly the evidence from ancient DNA, has generally not supported the multi-regional model in its original form, though the discovery of Neanderthal and Denisovan gene flow into modern human populations shows that the story is more complex than a simple, complete replacement of archaic populations by modern humans migrating from Africa. The process by which modern human populations spread across the globe was not a uniform migration. It involved multiple dispersal events, periods of population expansion and contraction driven by climatic changes and varying degrees of interaction with and replacement of previously established archaic hominin populations. Modern humans reached the Levant, the coastal region of the eastern Mediterranean, by at least 120,000 years ago, based on fossil evidence from Israeli sites. However, there is no evidence of a successful expansion from this early dispersal into the broader world. The populations responsible for the eventual colonization of Asia and beyond appear to have dispersed from Africa somewhat later, during a period of improved climatic conditions that may have facilitated movement through the region. Modern humans reached South Asia and Southeast Asia by approximately 60,000 to 70,000 years ago, and Australia by at least 50,000 years ago. The colonization of Australia represents one of the most remarkable dispersals in human evolutionary history, as it required crossing open ocean. The region between Southeast Asia and Australia involves multiple water crossings even when sea levels are at their lowest point during glacial intervals. The fact that people reached Australia so early implies that they had watercraft capable of open ocean travel, a technological achievement that would have required both the construction of appropriate vessels and the knowledge to navigate open water. The spread of modern humans through Europe occurred during a period when Neanderthals still inhabited the continent. Modern humans, referred to in the European context as anatomically modern humans or Cro-Magnons in older literature, appear in the European fossil and archaeological record from approximately 45,000 years ago. Within roughly 5,000 to 10,000 years of their arrival in Europe, the Neanderthals had disappeared. The cause or causes of Neanderthal extinction have been extensively debated. Possible factors include competition with modern humans for resources, the transmission of infectious diseases to which Neanderthals had no prior exposure, climate change, or some combination of these and other factors. The fact that modern humans did interbreed with Neanderthals shows that the two groups were not entirely reproductively isolated and did not consistently avoid contact. However, the scale of interbreeding was apparently limited, and the ultimate disappearance of Neanderthals as a distinct group was the eventual outcome. The arrival of modern humans in Europe is associated with what archaeologists call the Upper Paleolithic, a period characterized by a distinctive suite of cultural and technological innovations. Upper Paleolithic technology includes the production of long, thin blade tools from carefully prepared cores, the manufacture of a great variety of tools and implements from bone, antler, and ivory as well as stone, the production of standardized tools of specific types, and, most strikingly, the creation of art. The cave paintings of sites such as Lascaux in France and Altamira in Spain, dating to roughly 17,000 to 20,000 years ago, are among the most celebrated examples of prehistoric art. But older examples are known. The paintings of the Chauvet Cave in France date to approximately 36,000 years ago, and portable art objects, including figurines and ornaments, appear in the earliest Upper Paleolithic deposits. The production of representational art, images of animals, humans, geometric forms, and abstract symbols, is one of the most compelling indicators of the kind of cognitive and symbolic capacity that distinguishes modern humans from all other species. Creating an image of a bison or mammoth on a cave wall requires the ability to conceive of a representation as distinct from the thing it represents, to plan and execute a complex visual composition, and presumably to communicate something to an audience, whether a social audience of other humans or, as some have suggested, an audience conceived in terms of supernatural or spiritual beliefs. The existence of elaborate representational art in populations of modern humans from 36,000 or more years ago demonstrates that the cognitive capacities associated with fully modern human minds were present at least by this time. The cognitive capacities of modern humans differ from those of earlier hominins, including Neanderthals, in ways that are difficult to assess directly from the fossil record, but that are reflected in the archaeological record of behavior. Modern humans show evidence of planning depth, the ability to anticipate needs far in advance and prepare accordingly, that appears to exceed what is documented in archaic hominin sites. They show evidence of long-distance exchange networks, and through which raw materials and finished objects were moved over hundreds or even thousands of kilometers, implying social networks of a complexity and geographic scale not documented in earlier periods. They show evidence of cumulative cultural evolution, the ability to build on the innovations of previous generations in a way that allows technology and knowledge to become progressively more complex and effective over time, far exceeding what any individual could invent or discover alone. The anatomical changes that accompanied the emergence of modern Homo sapiens involved not only the increase in brain size that characterizes all later members of the genus Homo, but also changes in the organization and shape of the brain itself. The modern human skull is characteristically globular, rounded in shape, with a high domed brain case, a relatively flat face, a prominent chin, and the absence of the pronounced brow ridges seen in earlier hominins. These changes in skull shape reflect changes in the underlying brain. In particular, the parietal lobes, regions of the brain involved in integration of sensory information, spatial cognition, numerical reasoning, and aspects of language, are expanded in modern humans relative to archaic hominins, such as the Neanderthals. The development of language, the capacity for arbitrary symbolic communication through a structured system of sounds and rules, is likely to be among the most important cognitive innovations in the history of the human lineage. Language leaves no direct traces in the fossil record, as it is a behavior rather than a physical structure. However, certain anatomical features are relevant to the capacity for speech and language. The position of the larynx in the throat, the shape of the vocal tract, and the anatomy of the hyoid bone, a small bone at the base of the tongue that is related to vocalization, all influence the range of sounds that can be produced. The hyoid bone of Homo heidelbergensis and Neanderthals is similar to that of modern humans, suggesting these earlier hominins had some degree of vocal capacity. The anatomy of the tongue and larynx and the neural circuits controlling them are not preserved in fossils. Genetic evidence provides some clues. The FOXP2 gene, which plays a role in the fine motor control needed for speech, shows important sequence differences between modern humans and other apes. And the Neanderthal version of FOXP2 is identical to that of modern humans in the regions that differ from the chimpanzee version, suggesting that Neanderthals may have had some language capacity. However, the genetics of language are complex, and FOXP2 is only one of many genes involved. The expansion of the brain in the genus Homo over the past 2 million years is one of the most rapid and dramatic evolutionary changes recorded anywhere in the vertebrate fossil record. The brain of Homo habilis was approximately 600 cubic centimeters. The brain of modern Homo sapiens averages approximately 1,350 cubic centimeters. This is a more than doubling of brain volume in roughly 2 million years, a very short interval in geological terms. As brain tissue is metabolically expensive, the brain consumes roughly 20% of the body's total energy budget in modern humans, despite representing only about 2% of body weight. The expansion of the brain therefore required either an increase in the caloric quality or quantity of food, a reduction in the energy spent on other organs, or some combination of both. Richard Wrangham's cooking hypothesis, noted earlier, addresses the energy side of this equation. The reduction in the size of the gut in modern humans, which is smaller relative to body size than would be predicted from comparison with other primates, may represent the reduction of an energetically expensive organ that became possible when cooked food made digestion less demanding. The evolution of the brain is also intimately connected to the extended period of childhood and juvenile development in humans. Modern humans are born with brains that are far less developed than those of other primates at comparable developmental stages. A human infant is profoundly helpless compared to the offspring of other great apes at birth and remains dependent on adult care for an extraordinarily long time. This extended period of dependence is associated with the prolonged period of postnatal brain growth and learning that characterizes the human developmental trajectory. The extended childhood of humans provides a long window during which cultural knowledge, language, social norms, and technical skills can be transmitted from older to younger members of a social group. This extended period of learning may be one of the key biological foundations of the human capacity for cumulative culture. The social organization of hominin groups during the Middle and Upper Pleistocene is not directly observable, but it can be inferred from various lines of evidence. The size of campsites, the distribution of artifacts, and isotopic analyses suggesting movement between different environments all inform models of how groups were organized and how large they were. Modern hunter-gatherer societies, studied ethnographically, typically live in small bands of roughly 20 to 50 individuals that are part of a wider network of related bands maintaining regular social contact. This pattern of nested social groups, the small residential band embedded in a larger network, is thought to approximate the social organization of anatomically modern humans in the Pleistocene and may have deep roots in the social organization of earlier Homo populations. The transition from the Middle Paleolithic to the Upper Paleolithic in Europe and the equivalent transitions in other regions of the world are associated with a striking increase in the pace and complexity of cultural change. The rate at which new technologies and behaviors appear in the archaeological record accelerates dramatically. This is sometimes interpreted as reflecting a cognitive threshold, a point at which the neural substrate for fully modern cognition was in place, enabling a qualitative change in the capacity for cultural innovation and transmission. Alternatively, demographic factors, increases in population size and density that increase the probability that innovations will arise and be transmitted, may account for some or all of the observed acceleration in cultural change. These explanations are not mutually exclusive, and both cognitive and demographic factors probably played roles in the observed pattern. Throughout this long period of evolutionary development, the relationship between biology and behavior was bidirectional. Biological changes, increases in brain size, modifications of the vocal tract, changes in hand anatomy, enabled new behaviors. But behaviors also influenced biology through the selective pressures that different behaviors created. Cooking changed the selective environment for the digestive system. A social cooperation created selective pressures on social cognition and communication. The use of tools may have influenced the evolution of the hand and the neural circuits controlling fine motor movement. This interplay between biology and behavior over millions of years is one of the defining characteristics of human evolution and helps to explain why the human lineage shows such a distinctive pattern of change compared to other lineages. The Middle Pleistocene climate, the period from approximately 800,000 to 130,000 years ago, was characterized by a pattern of glacial cycles in which ice sheets advanced and retreated roughly every 100,000 years, a period that corresponds to one of the cycles in the Earth's orbital geometry. During glacial maxima, Bahati sea levels dropped substantially by as much as 120 meters at the most extreme points, exposing vast areas of continental shelf as dry land. Africa became somewhat cooler and drier. The Sahara Desert expanded. Forests contracted. These environmental changes created barriers to population movement and isolated human populations from one another, driving the geographic variation that can be observed in the fossil record of this period. During interglacial periods, conditions improved, barriers dissolved, and populations that had been isolated could come back into contact and intermix. The pattern of population isolation and secondary contact driven by climatic fluctuations has been important in shaping the genetic diversity of modern human populations. Much of the genetic variation seen among modern humans appears to have its roots in the structuring of African populations during the Middle Pleistocene. The population that ultimately gave rise to all living humans was not a single homogeneous group, but rather a set of partially isolated, interconnected African populations that periodically exchanged genes and that were exposed to somewhat different selective environments in different parts of the continent. This model of human origins, sometimes called the structured population model or the African multi-regional model, is gaining increasing support from both genetic and fossil evidence. The period from approximately 130,000 to 70,000 years ago encompasses the last interglacial period and the early stages of the last glacial cycle. During the last interglacial, when global temperatures were slightly warmer than today and sea levels were higher, modern humans were present in Africa and had begun to move into adjacent regions. As the climate cooled and the last glacial cycle commenced, populations experienced both opportunities and constraints. The cooling and drying of Africa may have reduced resources in some areas and pushed populations toward coastal zones and river valleys where food and water remained available. It is during this period that many of the behavioral innovations documented in southern and eastern African sites appear. Innovations that may reflect the adaptive responses of human populations under environmental stress. The bottleneck hypothesis, the idea that the human population underwent a severe reduction in size at some point during this period, perhaps around 70,000 to 75,000 years ago, has been linked by some researchers to the eruption of the Toba supervolcano in Sumatra. The Toba eruption, one of the largest volcanic events of the past million years, released enormous quantities of ash and aerosols into the atmosphere, potentially causing a period of global cooling lasting several years. Genetic data suggest that the effective population size of the ancestors of modern non-African humans was very small, perhaps as few as several thousand individuals, at some point in the relatively recent past, which is consistent with a severe population bottleneck. However, the link between the Toba eruption and a genetic bottleneck remains debated. And some archaeological evidence suggests that human populations in Africa and India survived the eruption with less disruption than would be expected from a catastrophic population crash. By approximately 70,000 years ago, modern human populations were beginning to spread more extensively out of Africa. The routes and timing of these dispersals are studied through a combination of ancient DNA, the genetics of living populations, and the archaeological record. One dispersal route appears to have gone through the southern Arabian Peninsula, possibly at a time when lower sea levels provided a land crossing of the Bab el-Mandeb Strait at the southern entrance to the Red Sea, and from there along the coast of South Asia toward Southeast Asia and ultimately Australia. Another dispersal event, somewhat later, moved populations through the Levant corridor into Central Asia, and from there into Europe and Eastern Asia. The spread of modern humans across the globe did not occur into empty landscapes. In addition to the Neanderthals in Europe and Western Asia and the Denisovans in Central and Eastern Asia, there were other hominin populations in different parts of the world. On the island of Flores in Indonesia, a diminutive hominin species called Homo floresiensis was present until approximately 50,000 years ago. Homo floresiensis had a very small body, approximately 1 meter tall, and a brain of just 380 to 420 cubic centimeters, smaller than that of Australopithecus. Despite this small brain, Homo floresiensis made stone tools and appears to have hunted pygmy elephants and other animals. The small body and brain of Homo floresiensis are thought to represent a case of island dwarfism, the evolutionary reduction in body size that occurs when populations of large animals are isolated on islands where food resources are limited. Whether Homo floresiensis descended from Homo erectus or from an earlier, small-brained hominin that dispersed to Flores before Homo erectus appeared is a question still under investigation. Another recently discovered hominin, Homo luzonensis, is known from fossils found on the island of Luzon in the Philippines and dating to approximately 67,000 years ago. Like Homo floresiensis, Homo luzonensis was small and appears to have been island adapted. These discoveries on Southeast Asian islands highlight the fact that the hominin family was considerably more diverse than was recognized even a few decades ago, and that the fossil record of hominin evolution in Asia remains incompletely known. The period at which modern humans spread across the globe was also a period of dramatic megafaunal extinctions. On nearly every continent and island that modern humans colonized, large mammals went extinct within a geologically short time of the arrival of people. In Australia, species including giant marsupials, enormous lizards, and large flightless birds disappeared. In the Americas, mammoths, mastodons, giant ground sloths, native horses, and a variety of other large animals vanished. On oceanic islands, extinctions followed human arrival almost universally. The close temporal correlation between human arrival and megafaunal extinction has led most researchers to conclude that human hunting was a primary cause of these extinctions, though the contribution of climate change, which was also happening during the late Pleistocene, is debated for some regions. The extinction of large animals had profound consequences for the ecosystems of the affected continents and islands, and also for the human populations who had depended on those animals as food sources. In regions where large prey animals disappeared, human populations had to adjust their economies, shifting toward the exploitation of smaller animals, plant foods, and marine resources. These economic adjustments were accompanied by technological and social innovations, the development of more efficient fishing and gathering technologies, for example, and changes in the size and structure of social groups. The end of the last glacial maximum, approximately 18,000 to 20,000 years ago, and the subsequent warming of the global climate during the Holocene epoch, beginning approximately 11,700 years ago, set the stage for the next major phase of human prehistory, which involves the development of agriculture and settled life. But before this transition is examined, it is important to note what had been achieved by the end of the Pleistocene in terms of the biological and behavioral evolution of our species. By the end of the Pleistocene, modern humans had colonized every habitable continent, lived in a wide variety of environments from tropical rainforests to Arctic tundra, and maintained ways of life based on hunting and gathering, supplemented by fishing, shellfish collection, and the exploitation of plant foods. They lived in social groups characterized by language, kinship systems, exchange networks, and accumulated bodies of knowledge transmitted across generations. They produced art, music, and personal ornaments. They buried their dead, often with grave goods, suggesting beliefs about an afterlife or the symbolic treatment of the dead. They had developed a diversity of stone tool technologies suited to different environments and tasks, as well as technologies based on bone, wood, fiber, and other organic materials. All of these achievements were the product of the evolutionary processes described in the preceding pages, the gradual accumulation over millions of years of biological and cultural changes that cumulatively transformed a small-brained, a tree-climbing ape into a species capable of colonizing the entire planet. The biological evolution of the human brain during the period covered in this second part of the documentary is accompanied by what might be called cultural evolution, the transmission and accumulation of knowledge, skills, and behaviors across generations through learning rather than through genetic inheritance. Cultural evolution is qualitatively different from biological evolution in several important ways. It can occur much faster than genetic evolution because information can be transmitted from one individual to many within a single generation. It is directed by the choices of individuals and groups, not purely by natural selection acting on random genetic variants. And it is cumulative in a way that biological evolution is not. Each generation can build on the knowledge accumulated by all previous generations, rather than starting from scratch. The development of language is central to the power of cultural evolution in the human lineage, as language vastly increases the efficiency and fidelity with which complex information can be transmitted between individuals. The long arc of biological evolution and the emergence of cultural evolution as an increasingly important force in human development converge toward the end of this second phase. By the beginning of the Holocene, the biological evolution of the human body and brain had largely reached the form seen in living people. The brain had achieved its modern size and organization. The skeleton had its modern proportions, and the anatomical machinery for language, fine motor control, and complex social behavior was fully in place. The subsequent history of the human species would be driven predominantly by cultural evolution, operating on a biological substrate that has changed relatively little over the past 50,000 years or more. The third phase of the human story begins as the last ice age draws to a close. The glaciers that had covered much of northern Europe, North America, and parts of Asia were retreating. Global temperatures were rising, sea levels were climbing as the enormous volumes of water locked in ice sheets returned to the oceans. Coastlines were shifting, forests were expanding into regions that had previously been tundra or open grassland. The human populations that had survived the last glacial maximum in refugia, areas where the climate remained tolerable even at the peak of glaciation, were beginning to expand and move into the landscapes opened by the retreating ice. But the Holocene epoch, the geological epoch that began approximately 11,700 years ago and continues to the present, is distinguished from the preceding Pleistocene by its relative climatic stability. While the Pleistocene was characterized by repeated, large amplitude swings between glacial and interglacial conditions, the Holocene has been a period of comparatively warm, stable climate. This stability, which is unusual in the context of the past several million years of Earth history, created conditions under which a way of life fundamentally different from the mobile hunting and gathering of the Pleistocene became viable. The way of life based on the deliberate cultivation of plants and the herding of domesticated animals, which is known as agriculture. The origins of agriculture represent one of the most consequential transitions in all of human history. Agriculture is not a single invention that occurred in one place at one time. It emerged independently in multiple regions of the world, in each case representing a gradual process of increased human manipulation and selection of wild plants and animals over long periods of time, eventually resulting in the domestication of species that became dependent on human management for their reproduction. The earliest well-documented center of agricultural origin is the region known as the Fertile Crescent, which encompasses parts of modern Turkey, Syria, Iraq, Lebanon, Israel, and Jordan, an area where a particular combination of environmental conditions and wild plant and animal species created favorable conditions for the development of farming. In the Fertile Crescent, the wild ancestors of the major crops of the ancient Near East, including emmer wheat, einkorn wheat, barley, lentils, peas, and chickpeas, grew in dense harvestable stands in areas of moderate winter rainfall. The wild ancestors of the major livestock animals of the region, wild goats, wild sheep, wild cattle, and wild boar, were also present. The hunter-gatherers who inhabited this region during the Natufian culture dating to approximately 14,500 to 11,500 years ago, were already extensively exploiting these wild resources. And there is evidence that some Natufian communities were semi-sedentary, returning year after year to the same locations to harvest wild cereals, long before the actual domestication of plant species. The domestication of wheat and barley, which involved the selection of individuals with traits that improved their usefulness to humans, larger seeds, non-shattering seed heads that do not release seeds before harvest, and other characteristics, occurred over a period of perhaps 1,000 to 2,000 years following the Natufian period. The domestication of goats and sheep followed a similar trajectory, as human populations began managing herds of these animals for meat, milk, wool, and hides. By approximately 9,000 to 10,000 years ago, fully agricultural communities were present in the Fertile Crescent, growing crops, raising livestock, and living in permanent or semi-permanent settlements. Agriculture spread from the Fertile Crescent into neighboring regions over the subsequent millennia. It spread westward into Europe, reaching the southeastern corner of the continent by approximately 8,500 years ago and spreading progressively northward and westward over the following several thousand years. It spread eastward into Central Asia and eventually into South Asia. The spread of agriculture was not simply the diffusion of ideas or practices from one group to another. It involved, at least in many cases, the actual movement of farming populations into new territories, as documented by ancient DNA analysis that shows the ancestry of European farmers differed substantially from that of the hunter-gatherers who preceded them. Agriculture also arose independently in several other parts of the world. In China, the cultivation of rice in the Yangtze River Valley and millet in the Yellow River Valley appears to have begun approximately 8,000 to 9,000 years ago. In the Americas, the cultivation of maize in Mesoamerica, potatoes and quinoa in the Andes, and squash and other plants in various regions began independently, also several thousand years ago. In sub-Saharan Africa, the cultivation of sorghum, millet, and other African crops was initiated by populations in the Sahel and savanna regions. In New Guinea, the cultivation of taro and other root crops appears to have a history extending back approximately 7,000 to 10,000 years. Each of these independent origins of agriculture reflects the independent discovery in different parts of the world of the same fundamental principle, that humans could increase the reliability and productivity of their food supply by actively managing plants and animals, rather than relying entirely on their wild abundance. The consequences of agriculture for human biology, health, society, and population size were profound and multifaceted. On the population side, agriculture dramatically increased the carrying capacity of the land, the number of people who could be sustained per unit area. Farming can produce far more calories per hectare than hunting and gathering in most environments, and it allows food to be stored in ways that buffer against seasonal and annual variability in food availability. As a result, agricultural populations grew rapidly compared to hunter-gatherer populations. The spread of farming populations across the globe was accompanied by a dramatic increase in total human population size, from an estimated few million people at the dawn of agriculture to tens of millions by the time the first cities arose. The health consequences of the transition to agriculture were, in many ways, negative in the short to medium term. Archaeological studies of skeletal populations comparing hunter-gatherers and early farmers in the same regions consistently show that early farmers had lower stature, reflecting poorer nutrition, higher rates of tooth decay from starchy diets, higher rates of iron deficiency anemia, and more evidence of infectious disease. The concentration of large numbers of people in permanent settlements, living in close proximity to domestic animals and their pathogens, oft created ideal conditions for the emergence and spread of infectious diseases. Many of the major epidemic diseases of human history, including smallpox, measles, influenza, and plague, appear to have their origins in pathogens that initially infected domestic animals and were transmitted to humans following the domestication of those animals. The dense, sedentary populations of agricultural societies provided the minimum population sizes necessary for these pathogens to sustain themselves, as epidemics require a continuous supply of susceptible individuals to maintain transmission. Despite these health costs, the agricultural populations grew faster than hunter-gatherer populations because of higher birth rates associated with the sedentary lifestyle and the ability to introduce supplementary foods to infants earlier than is possible in mobile hunting and gathering societies. The biological changes associated with the Neolithic transition, as the shift to farming is called in the archaeological literature, also include changes in the frequency of certain genetic variants in human populations. One of the best documented examples is the spread of genetic variants associated with the ability to digest lactose, the sugar found in milk, in adulthood. Most mammals lose the ability to digest lactose after weaning, as the gene encoding the lactase enzyme is normally switched off at that time. In populations where dairying, the use of milk and milk products from domestic animals, became an important part of the economy, variants that maintained lactase activity into adulthood were strongly favored by natural selection, as they allowed individuals to extract calories from a reliable and abundant food source. These variants reached high frequency in European and some African populations over a period of roughly 5,000 to 7,000 years, one of the most rapid and well-documented examples of natural selection in the recent history of the human species. The development of agriculture was followed in some regions by the development of increasingly complex social organization, the accumulation of material wealth and its unequal distribution, specialization of labor, and the rise of the first cities and states. But these developments, which historians sometimes refer to collectively as the development of civilization, are not biologically driven in the sense of requiring any further genetic change in the human population. They are products of cultural evolution operating on the biologically modern human brain and body that had been in place for tens of thousands of years before agriculture appeared. The capacity for complex social organization, symbolic communication, planning, cooperation, and technological innovation, all of which are prerequisites for the development of urban civilization, was already fully present in the hunting and gathering populations of the late Pleistocene. What changed with agriculture was not human biology, but the social and economic context in which these capabilities were expressed. But the first cities appeared in Mesopotamia, the region between the Tigris and Euphrates rivers in modern Iraq, approximately 5,500 to 6,000 years ago. Cities require a reliable food surplus, enough food produced by farmers to feed not only the farmers themselves, but also a population of craftspeople, administrators, soldiers, priests, and others not directly involved in food production. They require systems of storage and distribution. They require some form of political organization capable of coordinating the activities of large numbers of people and managing conflicts. And they require systems of record keeping, which, in Mesopotamia, took the form of writing, developed approximately 5,200 years ago initially for accounting purposes, and later for a much broader range of communication. The development of writing is another one of the great transitions in human cultural history. Writing allows information to be stored outside individual human memories, transmitted across distances and time with greater fidelity than oral tradition allows, and accumulated over generations in a way that enormously extends the reach of cultural memory. The development of writing in Mesopotamia, using wedge-shaped marks on clay tablets, occurred independently of the development of writing in China, which appears to have begun approximately 3,000 years ago, and in Mesoamerica, where the earliest writing systems appear several thousand years ago as well. In each case, writing arose in the context of complex societies with an administrative need to track resources, obligations, and transactions. The development of cities and states brought with it new forms of social inequality. In hunter-gatherer societies, while individuals differ in status and influence, the accumulation of substantial material wealth by some individuals at the expense of others is generally constrained by the mobile lifestyle and the cultural norms of sharing that characterize most such societies. Agriculture allowed wealth to accumulate in the form of stored grain, livestock herds, and land. Once wealth could accumulate, it could be transmitted to offspring, creating hereditary social classes. The skeletal record of early agricultural and early urban societies shows clear evidence of differential access to nutrition and health care. Individuals from higher status contexts tend to show better nutrition, fewer signs of infectious disease, and less physical evidence of heavy labor than lower status individuals from the same communities. The biological evolution of Homo sapiens has not stopped since the emergence of anatomically modern humans. Natural selection continues to operate on human populations, and there is evidence from population genetic studies that a number of human genes show signatures of recent positive selection, meaning that certain genetic variants have increased in frequency in specific populations over the past few thousand to tens of thousands of years because they conferred a fitness advantage in a particular environment. Some of these variants are related to disease resistance, diet, and skin pigmentation, the reduction of skin pigmentation in populations that move to high latitudes where sunlight is less intense. It is one of the best documented cases of recent human evolution, driven by the need to synthesize adequate vitamin D in conditions of reduced UV exposure. However, the pace of biological evolution in the human lineage over the past 50,000 years has been much slower than the pace of cultural change, and the dominant story of the past 10,000 years is one of cultural evolution rather than biological evolution. The anatomical structure of modern humans reflects a history of evolutionary pressures and trade-offs accumulated over millions of years. The upright posture that was adopted by the hominin lineage several million years ago created a set of structural challenges that persist in the modern human body. The shift to bipedalism required a narrowing of the pelvis relative to the broad, shallow pelvis of other apes, due to improve the mechanical efficiency of walking. But this narrowed pelvis created a conflict with the evolution of a large brain. As brain size increased, the head of the newborn became larger, and the birth canal remained constrained by the demands of efficient bipedalism. The result is that human childbirth is considerably more difficult and dangerous than in other primates, and human infants are born at an earlier developmental stage, more helpless than the infants of other great apes. This obstetric dilemma, as it is sometimes called, has shaped many aspects of human reproductive biology and social organization, including the degree to which cooperative care of infants by multiple individuals beyond the mother is a universal feature of human societies. But the lower back pain that affects a large proportion of the adult human population is another legacy of the evolutionary transition to upright walking. The lumbar spine of humans is under considerable compressive load in an upright posture, and the intervertebral discs, the cartilaginous structures between vertebrae that absorb shock and allow movement, are vulnerable to herniation, degeneration, and other forms of mechanical failure. The structure of the human lumbar spine reflects compromises between competing demands that have never been perfectly resolved over the course of human evolutionary history. The immune system of modern humans reflects a history of exposure to a particular set of pathogens in particular environments, and this history has shaped patterns of immune function that are sometimes poorly suited to the environments in which many people now live. The hygiene hypothesis, which has been refined and extended in subsequent research, proposes that the dramatic increase in the prevalence of allergic and autoimmune diseases in industrialized populations over the past century is partly attributable to reduced exposure in early childhood to the parasites, bacteria, and other microorganisms to which human immune systems were adapted over evolutionary time. The immune system, calibrated by millions of years of exposure to a particular suite of pathogens and parasites, may be prone to misdirected inflammatory responses when deprived of its co-evolved microbial partners. The human brain, now shaped by millions of years of evolution in environments and social contexts very different from those of modern industrialized societies, exhibits various tendencies and biases that reflect adaptations to ancestral conditions rather than to the current environment. The tendency to prefer calorie-dense foods, fats and sugars, reflects an environment in which such foods were rare and valuable, not one in which they are abundant and cheaply available. The tendency toward anxiety in social situations reflects the importance of social relationships and social standing in small group hunter-gatherer societies, where ostracism could be life-threatening. These mismatches between evolved human psychology and the modern environment are studied extensively in the field of evolutionary psychology, with the goal of understanding the biological roots of human behavior and mental health. The study of human evolution has itself evolved considerably over the past century and a half. When Charles Darwin published On the Origin of Species in 1859 and The Descent of Man in 1871, the fossil evidence for human evolution was virtually non-existent. Darwin's arguments for human evolution from earlier primates were based primarily on comparative anatomy, embryology, and biogeography. The subsequent 150 years have seen an extraordinary accumulation of fossil evidence, refined dating techniques, comparative genomics, ancient DNA analysis, and sophisticated archaeological methods that have transformed our understanding of the human past. The field of paleoanthropology, the study of human evolution through the fossil record, has moved from the interpretation of a handful of fragmentary specimens to the analysis of dozens of hominin species known from thousands of individual fossils from sites across Africa, Europe, and Asia. Dating methods including radiometric dating, paleomagnetic dating, and a variety of other techniques allow researchers to determine the age of fossil-bearing sediments with considerable precision. Stable isotope analysis of fossil bones and teeth provides information about the diet and habitat of ancient hominins. Ancient DNA extracted from well-preserved specimens, a technology that has revolutionized the field since the mid-2000s, provides direct genetic evidence of relationships between populations and of interbreeding events. Geometric morphometrics, the precise three-dimensional analysis of the shape of skulls and other bones, allows quantitative comparisons between specimens and populations that supplement traditional qualitative description. The genomics of living human populations has also contributed enormously to understanding human evolutionary history. The analysis of the genetic variation carried by living people in different parts of the world provides evidence about the history of population movements, expansions, contractions, and admixture events. The greater genetic diversity of African populations compared to non-African populations, a consistent finding across numerous studies, is consistent with the African origin of modern humans and the bottleneck effect associated with the spread of a subset of the African population into the rest of the world. The patterns of genetic variation within Africa reflect the complex history of population structure and exchange on the continent over hundreds of thousands of years. The patterns of variation between African and non-African populations and among non-African populations themselves provide a record of the dispersal routes, population sizes, and contact events that characterize the global spread of modern humans. The relationship between genetics and culture in human evolution is a topic of ongoing interest and debate. Cultural practices can influence genetic evolution by changing the selective environment. The development of dairying drove the evolution of adult lactase persistence, as noted earlier. Conversely, genetic differences between populations can influence cultural evolution. If they affect the range of learning abilities, social tendencies, or other traits that influence what kinds of cultural practices are developed and maintained. The question of the extent to which differences between human populations in cultural practices, social organization, or cognitive abilities might reflect genetic differences is a sensitive one, touching as it does on long histories of scientific racism and misuse of biological concepts. The scientific consensus, supported by extensive genomic and psychological research, is that the genetic differences between human populations are small relative to the within-population variation, that there is no evidence for meaningful genetic differences in cognitive ability between populations, and that the observed differences in cultural practices and social organization between human societies reflect the enormous power of cultural evolution and historical contingency rather than genetic differences. The deep time perspective provided by the study of human evolution reveals several important truths about the nature of our species that are not apparent from shorter-term perspectives. One is that the traits we think of as distinctively human, upright walking, large brains, language, tool use, complex social life, did not appear simultaneously as a package, but accumulated gradually over millions of years, not with different components appearing at different times and in different combinations. Another is that the human lineage has never been a single linear progression from primitive to advanced, At virtually every point in the past 7 million years, multiple hominin species existed simultaneously, occupying different regions and exhibiting different combinations of traits. The survival of the particular lineage that led to modern humans and the extinction of all other hominin lineages was not predetermined or inevitable. It resulted from the particular combination of biological traits and behavioral capacities that our lineage possessed, from the particular sequence of environmental changes that occurred, and from a measure of contingency, of events that could have gone differently. A third truth, revealed by the deep time perspective, is the antiquity and universality of human behavioral capacities that might otherwise seem unique to particular cultural traditions. Art, burial of the dead, the use of pigments for body decoration, long-distance trade, and the use of complex tools are all documented in anatomically modern humans living under very different cultural and environmental conditions, and in widely separated parts of the world, suggesting that these behaviors are expressions of a shared human capacity rather than inventions of any particular culture or tradition. The universality of certain behavioral patterns across all known human societies, including language, music, marriage, kinship systems, and moral norms, points to deep biological roots in the evolved human mind. Human biological diversity, that as it exists today, reflects tens of thousands of years of evolutionary differentiation in different environments following the dispersal of modern humans from Africa, combined with the gene flow between populations that has always been a feature of human population history. The differences between human populations in visible traits such as skin color, hair texture, and facial structure are real but superficial in a biological sense. They involve a very small fraction of the total genetic variation in the species. They arose relatively recently in evolutionary time in response to specific regional selection pressures such as UV radiation and temperature, and they do not correspond to meaningful differences in cognitive ability, temperament, or other traits. The concept of race as a biological category has been largely rejected by geneticists and physical anthropologists because the pattern of human genetic variation does not support the existence of discrete biological races. Human populations grade into one another geographically. The boundaries between any putative races are arbitrary, and most genetic variation is found within rather than between populations. The evolutionary origins of language, one of the defining features of the human species, remain a topic of intense research and considerable uncertainty. Language is a uniquely human trait in its full modern form. No other animal has a communication system with the same combinatorial complexity, the same capacity for expressing an unlimited number of novel meanings, or the same degree of recursion, the ability to embed phrases within phrases, as human language. The question of when language in its modern form arose and what evolutionary pressures drove its development is difficult to answer from the fossil record alone. Some researchers argue that fully modern language appeared relatively recently, perhaps only 50,000 to 70,000 years ago, coinciding with the apparent acceleration of cultural complexity in the Upper Paleolithic. Others argue that language has a much deeper history in the hominin lineage, and that the behavioral evidence for complex cognition extending back several 100,000 years reflects linguistic capacity. The genetic evidence, including the FOXP2 gene and other loci associated with language-related processes, does not yet provide a definitive answer to this question. The evolution of human cognition and behavior also involved the evolution of emotion, empathy, and social attachment, capacities that are deeply rooted in the mammalian brain and that have been elaborated and modified in the primate lineage. The capacity for empathy, for modeling the emotional and mental states of other individuals, is found in other primates and even in some non-primate mammals, but is particularly well developed in humans. Empathy is the biological foundation for the moral concern for others that is a universal feature of human social life. The evolution of enhanced empathy in the human lineage is thought to be associated with the increasing demands of cooperative social life, the extended care of infants and children, and the benefits of social coordination in groups. The neural circuits underlying empathy and social cognition in humans involve regions of the prefrontal cortex that are particularly enlarged relative to other brain regions in modern humans compared to other primates. The biological basis of human social behavior, cooperation, altruism, conflict, and competition, is studied from several perspectives within evolutionary biology. Inclusive fitness theory, which takes into account not only an individual's own reproduction, but also the reproduction of relatives who share genes, explains the evolution of altruistic behavior toward kin. Reciprocal altruism, the exchange of benefits over time between individuals who are not necessarily related, provides a framework for understanding cooperation among non-kin. The theory of cultural group selection proposes that groups whose members cooperate more effectively outcompete groups with less cooperation, driving the evolution of norms and institutions that promote within-group cooperation. Each of these frameworks contributes to understanding aspects of human social behavior, and it is likely that multiple mechanisms have been important in shaping the particular form that human sociality takes. The transition from a predominantly biological evolutionary framework to a predominantly cultural one over the past 50,000 years or so has not eliminated the role of biology in human behavior and society. Human social institutions, family structures, economic systems, legal systems, political organizations, religious traditions are all built on a biological foundation of evolved psychological tendencies, emotional responses, and cognitive capacities. Understanding the biological underpinnings of these tendencies and capacities is valuable not because it determines or constrains what social institutions are possible, but because it helps to explain why certain patterns recur across diverse cultures and why certain institutional designs are more stable or more conducive to human flourishing than others. The field of evolutionary medicine applies the principles of evolutionary biology to understanding patterns of human health and disease. The concept of evolutionary mismatch, the idea that many common diseases and health problems reflect mismatches between the environments and behaviors for which our biology evolved and the environments and behaviors of modern life, has proven productive in generating hypotheses about the causes of conditions including obesity, type 2 diabetes, cardiovascular disease, myopia, and various mental health conditions. The recognition that the human body and brain were shaped by selection. In environments very different from those of modern industrialized societies, provides a framework for understanding why so many common conditions are associated with aspects of modern diet, physical activity patterns, sleep habits, and social environments. The study of ancient DNA from archaeological populations has opened a new window onto the demographic history of human populations over the past 50,000 years. Ancient DNA studies have revealed that the current genetic composition of populations in various parts of the world is the product of a complex series of migrations, admixture events, and population replacements, often departing significantly from what had been inferred on the basis of the modern genetic diversity alone. In Europe, for example, ancient DNA studies have shown that the genetics of modern Europeans reflects the contributions of at least three distinct source populations: the indigenous hunter-gatherers of Paleolithic Europe, the Anatolian farmers who spread into Europe during the Neolithic, and a population from the Pontic steppe, the grasslands north of the Black Sea, that spread westward into Europe during the Bronze Age, approximately 4,000 to 5,000 years ago. Similar complexities have been revealed in the histories of populations in South Asia, East Asia, the Americas, and elsewhere. The Bronze Age migrations from the Pontic steppe, associated archaeologically with the Yamnaya culture, appear to have spread not only genes but also languages. The Indo-European language family, which includes most of the languages of Europe, Iran, and South Asia, is now thought by many linguists and geneticists to have been spread in large part by the movement of Yamnaya-related populations. This migration, occurring within the past 5,000 years, was among the most well-documented examples of a demographic event that simultaneously reshaped the genetic and linguistic landscape of large parts of the world. It illustrates that even in the relatively recent human past, well within the period of recorded history in some regions, major demographic events were occurring that profoundly shaped the populations and cultures of today. The study of human remains from archaeological contexts raises ethical questions that have become increasingly prominent in recent decades. Many of the skeletal collections that have been central to the study of human evolution and bioarchaeology were assembled during periods when the rights and interests of the communities from which the remains came were not taken into account. That indigenous communities in many parts of the world have advocated for the repatriation of ancestral remains and associated objects from museum and university collections. These claims reflect deeply held cultural and ethical convictions about the proper treatment of the dead and the ownership of cultural heritage. The scientific community has been in ongoing dialogue with indigenous communities about how to balance the scientific value of studying ancient human remains with the rights and interests of the communities with which those remains are affiliated. This dialogue has led to significant changes in the practices of paleoanthropology and bioarchaeology, including legal frameworks for repatriation in several countries and the development of collaborative research models in which indigenous communities are partners rather than subjects of study. The impact of human evolution research on broader social and philosophical questions has been significant. The recognition that humans are the products of the same evolutionary processes that shaped all other living organisms, that there is a continuity between human life and all other life, has implications for how we understand our relationship to the natural world. The evolutionary perspective has contributed to the understanding that many features of human behavior that might previously have been explained purely in terms of cultural norms, rational choice, or spiritual dictates have deep biological roots. This does not eliminate the importance of culture, choice, or ethics, but it adds a layer of understanding about why certain patterns of behavior are so pervasive and so difficult to change. The evolutionary history of the human species is also relevant to understanding the current situation of our species on this planet. The capacity to dramatically modify environments, which has been a feature of the human lineage since at least the use of fire for habitat management by early Homo sapiens, has scaled up enormously with the development of agriculture, industry, and modern technology. The ecological footprint of the human species, the total impact of human activity on the biosphere, is now large enough to be visible in the geological record, prompting geologists and earth scientists to consider whether the current interval of Earth history should be recognized as a new geological epoch, the Anthropocene, distinguished by the pervasive influence of human activity on global biogeochemical systems. The evolutionary framework helps to explain why the human impact on the biosphere has been so large and so rapid. The combination of large brains, cumulative cultural evolution, cooperative social organization, and language gave Homo sapiens a capacity for technological innovation and social coordination that no previous species possessed. This combination allowed human populations to extract resources from the environment and to transform ecosystems at rates far exceeding those associated with any other species in the history of life on Earth. The agricultural revolution, the industrial revolution, and the information revolution are successive accelerations of the same underlying process: the application of culturally accumulated knowledge to the manipulation of the natural world. The pace of cultural evolution in the human species has been accelerating over the course of recorded history, and the pace of acceleration has itself been increasing. Agricultural societies developed over thousands of years. The Industrial Revolution unfolded over centuries. The digital information revolution has occurred over decades. This accelerating pace of change creates a situation in which the biological substrate of the human mind, shaped by millions of years of evolution in environments where change was slow, is confronted with a pace of social, technological, and environmental change that has no precedent in the evolutionary history of the species. The psychological challenges associated with rapid social change, including disruption of established social roles and identities, information overload, and the erosion of traditional community structures, are among the subjects of ongoing research in evolutionary psychology and related fields. Reflecting on the full span of human evolutionary history, from the first hominins walking bipedally across the African savanna more than 6 million years ago, to the anatomically and behaviorally modern humans who eventually populated every habitable corner of the planet, several themes emerge that deserve emphasis. The first theme is deep continuity. The human body and mind are the products of an unbroken chain of evolutionary history, extending back not just millions of years to the first hominins, but hundreds of millions of years to the first vertebrates, and ultimately to the origin of life itself. Every system of the human body, the skeleton, the nervous system, the immune system, the reproductive system, contains structures and mechanisms that were inherited from distant ancestors and modified, sometimes dramatically, over vast spans of time. Understanding the human body in the context of its evolutionary history provides insights into its design and its limitation that no other framework can provide. The second theme is the interaction of biology and culture. The humans are simultaneously biological organisms shaped by natural selection and cultural beings whose behavior is profoundly influenced by learned knowledge, values, and traditions. These two aspects of human existence are not in opposition, they are deeply intertwined. Biology shapes the capacities within which culture operates. Culture creates selective environments that shape biology. The distinction between nature and nurture that has sometimes been drawn sharply in popular discussions of human behavior is fundamentally misleading. In the human case, nature and nurture are inseparably intertwined, each shaping the other across evolutionary and developmental time. The third theme is contingency. The particular form that human evolution took, the specific sequence of anatomical, cognitive, and behavioral changes that led to modern Homo sapiens, was not predetermined. It resulted from the interaction of biological variation, environmental conditions, and historical accident. Had the last ice age been slightly different, had the African climate of 2 million years ago followed a different trajectory, had any number of other events in the history of life on Earth gone differently, the particular configuration of traits that characterizes modern humans might not have arisen. The human species, like all species, is a product of history, a particular outcome of a process that could in principle have produced many different results. The fourth theme is the interconnectedness of life. The human evolutionary story cannot be told in isolation from the broader story of life on Earth. The emergence of hominins was possible only because of 500 million years of vertebrate evolution that preceded it. The characteristics of early hominins were shaped by the plants, animals, and environments they lived among. The extinction of the non-avian dinosaurs opened ecological space that mammals, including the primate ancestors of humans, filled. The domestication of plants and animals that launched the agricultural revolution was possible only because millions of years of co-evolution between plants and their animal pollinators, seed dispersers, and grazers had produced species with characteristics that humans could exploit and selectively modify. Humans are part of the web of life, not separate from it, and the evolutionary history of our species is embedded in and inseparable from the broader evolutionary history of all life on this planet. The fifth and final theme is the ongoing nature of evolution. The human species is not a finished product. Natural selection continues to operate on human populations, as it does on all biological populations. Cultural evolution continues to transform human societies at a pace that far exceeds biological evolution. The relationship between the human species and the other species of the biosphere, a relationship that has been characterized by an increasing human impact on the rest of the living world, continues to evolve. The future evolutionary trajectory of the human species, including the effects of modern medicine and technology on natural selection, the potential impacts of deliberate genetic modification, and the long-term consequences of the ecological transformations driven by human activity, are questions of genuine scientific interest that lie beyond the scope of the historical narrative told in this documentary. What can be said with confidence is that the human species, as it exists today, is the product of a process of evolutionary change that has been ongoing for billions of years and that is produced through the accumulation of countless small changes, a form of life with remarkable and unprecedented capabilities. The capacity for language, for abstract thought, for cooperative endeavor on a global scale, for the accumulation and transmission of knowledge across generations. These are not miracles or accidents, but the outcomes of natural processes operating over geological time on a planet that has proven, through its long history, to be an extraordinarily productive environment for the evolution of complexity. The story of human evolution is the story of how one thread of life on Earth following the same principles that govern all life, arrived at a particular set of capabilities and characteristics. And that story, in its depth and its richness, is one of the most illuminating perspectives available for understanding what human beings are and how we came to be here.